Okay, to start today, I want to go through the system that regulates metabolism, and then we're going to have you think through it and practice a bit for yourself, and I'll look back at a couple of the homework problems, and we'll talk about how to solve those homework problems. Then move on to more on the endocrine system. Remember, we have an exam on Wednesday, a week from Wednesday. Okay, so we have about 2 more lectures on the endocrine system, and then we'll have a couple on reproduction, which really just follows logically from the endocrine system because it's all controlled by neural hormones. So we'll talk about reproduction, fertility, and then exam 3 a week from Wednesday. So I hope this is getting familiar to you. What I really wanted to do was run through it really quickly and then give you guys a chance to ask questions. So hypothalamus here, pituitary gland, thyroid gland. And I want you thinking about what the hormones are from the hypothalamus. It's what hormone? Okay, TRH. And pituitary, TSH. And thyroid gland, okay, T3 and T4. T3 and T4 are used interchangeably with thyroid hormones. People use both terms. And thyroxine is commonly used for T3 as well. We're going to run into a few more cases where there are multiple names for exactly the same thing, so we'll talk about that occasionally. I wish it weren't that way because it just adds to the confusion, but there it is. Okay, so we have information from the body talking about what are conditions in the body. Are you cold? Is there adequate food or inadequate food? And other factors like changing season for lots of animals. Reproductive state changes metabolic rate for a lot of animals. There are different receptors for those. In some cases, it could be the TRH neurons themselves. In other cases, they may be getting input from other neurons. We aren't going to care which. What I'm going to want you to do is be able to come up with hypotheses for how these could be controlled. One of the questions in the homework was related to that, and that's the kind of thing I might put on an exam. So at this point, you know that you could make a G-protein coupled receptor or an ionotropic, so metabotropic or ionotropic receptor respond. No matter whether it's neurotransmitter release or some direct sensory input. So it could be that the TRH neurons themselves have thermoreceptors. They're detecting, in that case, temperature in the brain. Okay? Or it could be they're getting information from sensory neurons that are coming from other parts of your body, telling you, for example, is your core too cold? In which case, you might want to increase metabolic rate to warm up. So that has to be able to start an action potential. So the same things we were talking about in sensory neurons. It's exactly the same toolkit, exactly the same proteins. You need to get enough charge to the spike initiating region to start an action potential. And when you get down to the end of that TRH neuron, there has to be vesicles of what would have been neurotransmitter if this was a neuron releasing a chemical message to another neuron, but in this case, it's a neuron that's just releasing its message outside of the cell in the extracellular space near a capillary. If it's near a capillary, we'll talk many times about the fact that capillaries are leaky. They allow molecules out. Molecules can also diffuse in if they're going down their concentration gradient. So release TRH here right outside a capillary. A lot of it is going to wind up inside the capillary and be carried away. In this case, that capillary is very close to the base of the hypothalamus, and it's very close to the anterior pituitary gland. So there's a direct connection. It's called a portal blood vessel. so that whatever is released here winds up near cells that secrete TSH in the anterior pituitary. So these little blue dots represent the TRH. And it binds to TRH receptors in the anterior pituitary. We'll talk a bit more about portal vessels a time or two later on. Portal vessels are unusual in that blood, there's a set of capillaries that can pick something up in one location, in this case, base of the hypothalamus from TRH neurons, and instead of going to a vein and back to the heart and then out to the rest of the body, they go from a capillary to a slightly larger vessel, or quite a bit larger in some portal systems. Anyway, capillary to a larger vessel, and then another capillary. So what portal vessels do is they pick things up in one location and they deliver it somewhere else. Your biggest portal system picks up the newly digested materials from your intestine and delivers them to the liver, where the liver does some pre-processing, it detoxifies things, it checks for, it's a major immune organ, it checks for viruses and bacteria and attacks them if they're present. So if, for example, you got a bit of sharp stuff or grit or whatever in your food and it led to small cuts in the intestine, which has to happen all the time, bacteria and viruses can get in, your liver attacks whatever gets in and kills them. So a portal vessel delivers stuff from your intestine straight to your liver, meaning that you don't deliver all those bacteria and viruses through all the rest of the body before your liver has a chance to attack them. In this case, the portal system picks up a molecule that's locally abundant but would be diluted tremendously in the rest of the body and delivers it straight from the hypothalamus down to the anterior pituitary. Okay, that results in TSH release. So what's TSH doing? Well, TSH is binding to its receptor. So if this was TSH, it's binding to its receptor. That could be a G protein coupled receptor. It does produce a second messenger. So let's just hypothesize that it is. The second messenger might be cyclic AMP. That second messenger goes over and activates a cotransporter. We talked about cotransporters of sodium and glucose, sodium and amino acid, but there are lots of other cotransporters. This one concentrates iodine in cells of the thyroid gland. Okay, so we're in cells of the thyroid gland, so we have all this iodine inside, and the second messenger also activates the enzymes that convert an amino acid into T3 or T4. Remember, T4, 4 iodines on it, T3, 3. So we wind up with T3 and T4. Okay, we can just call them T4 and T3. I do want you to remember that T3 is the active one. If you like the idea of remembering tetraiodothyronine and triiodothyronine, you're welcome to do that too. There's a negative feedback pathway. If metabolic rate gets too high, so for example, injury, damage, receptors not responding properly in the thyroid gland could result in excessive secretion of T3 and T4. And excessive secretion of T3 and T4 causes Graves' disease. It's or hyperthyroidism. Hyper is for a lot. Hypo is too little. Hyperthyroidism. I had a friend who developed hyperthyroidism at about age 22 or something like that, 23. And she wound up with some anxiety, hungry all the time, lost weight. Her resting heart rate got up to something like 120, so sitting there like you guys are, 120 beats a minute. She was originally diagnosed as just having stress. She had just recently gone through a really bad marriage and a bad divorce, and maybe she was about 24, I can't remember. I don't think she was married quite that young. But anyway, and so her doctor said, it's just stress. And she was saying, No. The stress was before I got divorced. Now I feel great. And finally went to an endocrinologist who immediately put her on an IV and started treating her instantly. He said her heart rate at that point, I think it was something, resting heart rate was getting up to around 130 or 140, which is quite risky. You're getting to the point where your heart can't actually beat that fast, and so it loses its synchronous beat. So you need a negative feedback pathway. So here is a negative feedback pathway. This line means inhibition. And if the thyroid gland is overactive, you can get some suppression. If it doesn't suppress it enough, you wind up with Graves' disease. How does that work? Well, it's actually, the T4 is what's doing the inhibition, but it's T3 that is acting. And I've shown a separate cell converting T4 to deiodinase, but it could be going on inside the TRH neuron itself. Okay, so T4 getting converted by this enzyme, deiodinase, DIO, you don't need to remember the name, but there is an enzyme that does it, gets converted to T3, and then that T3 inhibits TRH. Okay, so it inhibits TRH secretion. So that's the regulation. Cold, excess food, other things will increase metabolic rate. You are right now, as you spend more time cold than you were during the summer, that's actually triggering every moment, every time you're a little bit cold with your core body temperature, that's actually triggering an increase in metabolic rate. I used to notice this particularly when I was at Michigan. Michigan, when the temperature got down to around 30 in the winter, everybody was bundled up. As soon as it hit first day above freezing with a bit of sun in the spring or even late winter, and there were people out in shorts and t-shirts. It's 32, 33, 35 degrees, they're out in shorts and t-shirts, and they're not cold. The thing is, their metabolic rate has increased, and so they're staying warm because of that. Okay, what does it do? First of all, T3 and T4 are not all that soluble in blood, and so you have binding proteins. So binding proteins are made in the liver, and that's what these little squiggles are supposed to represent for T3 and T4. So the T4 and T3 are constantly popping off and on these binding proteins, but at any given time, many more of them are held by these binding proteins than are free. The free ones can act. The T3 can simply get into any cell that has a transporter, bind to the receptor. The receptor is a transcription factor. I'm showing it as a single protein, as a monomer. It's actually a dimer. They're two identical subunits, and it's only active with the T3 bound to both. But those can bind to any site that fits a T3 receptor, and those can be repressors, repressor binding sites, in which case the gene is not expressed. But T3 and its transcription factor, its receptor, have the potential to be both enhancers or repressors, like other transcription factors. So if the binding site is an enhancer for that gene, so T3 and its receptor will turn down genes, reduce the expression of genes that are useful for conserving energy and for having lower metabolic rate, and it'll enhance transcription of genes for proteins that help use energy. Okay. And so you wind up with transcription. I've shown what the enhancers do here. I just called them protein 1, protein 2, and protein 3. Actually, it's quite a few proteins. What do they do? Well, thyroid hormone controls a lot of things, so it's not some small set of things. but one of them is converting glycogen to glucose. So here I'm showing a glycogen. Glycogen is a polymer of glucose molecules, so it's multiple glucose molecules in a branching pattern. So that would be glycogen converted to individual glucoses. It converts fats to free fatty acids. Okay, so it's using up fats, converting them to a useful source, and then protein synthesis. It stimulates protein synthesis, so you make new proteins. All of those act to make your use of energy faster. Your body temperature is going to be higher because you're generating more heat. And whatever things are going on are going to go on faster. Okay, and then locally I mentioned that cells that either themselves can convert T4 to T3 or have nearby neighbors that can, they can increase metabolic rate just for those particular cells. So the result is you can have some cells that are functioning at a fairly low rate, but others that you need to enhance, say immune system cells during an infection, could potentially convert T4 to T3 and use it to increase their metabolic rate. So they can be more active under those conditions. So you could imagine, we could propose that immune system cells, when they're inactive, when you have no infection, none of those cells or any of the nearby ones are producing the deiodinase that can convert T4 to T3, and they have a low metabolic rate. There's nothing for them to do. But have an infection, and some chemical message related to the infection could induce the production of the deiodinase that converts T4 to T3. And now you have upregulated metabolism for those cells, so they now become more active. You have to be able to turn this off. So you have clearance. It's a concept for all hormones, actually also a concept for drugs, including toxins. For any kind of medicinal drug we take, it's really important that research gets done on clearance rates. How fast is it gotten rid of? Because you need to know that before you can establish a dosing pattern. Okay. And clearance takes two forms. One is enzymatic conversion. So an enzyme that will inactivate them, and it converts T3 into T2, takes off one of those iodines, and T2 cannot bind to the receptor. And it converts T4 into reverse T3. It's not actually reverse, but the active form of T3 has one particular iodine that is missing from the 4 sites where iodine can bind. And reverse T3, it's a different iodine. Okay, so it converts T4 into a form of T3 that can't bind the receptor. The other is excretion, so blood that goes to your kidney, and in your kidney, the excretory tubules we'll talk much more about this later. They're tiny gaps in the beginning of the nephron, and so all of these get in, and they're excreted. Okay, now I want you to spend a moment going through this whole thing in your mind. Close your eyes and see if you can go through all the steps. You can draw in the air if you want to. You can look up when you need to. I'll try and flip this back and forth between the two every now and then. This is a useful study technique if you don't already do it. Okay, so look when you need to, but close your eyes and try and picture the whole thing in your head. You can make the diagrams more simple if you want. Once you're able to go through it, check your understanding with your neighbor. So if there's anything you're not clear on, how it happens step by step by step, and why. Okay, let me try an eyes closed exercise. I want to get a sense of how many people feel they understand this pretty well. They don't have major questions about it. So close your eyes. Close your eyes closed. Some of you have your eyes open. I'm seeing some people with eyes still open. Close your eyes. Still multiple people with eyes open. Close your eyes. Okay, eyes closed. Okay, eyes closed. Okay, raise your hand if you feel like I still have some gaps in here and I have some questions. Okay, hands down. You can open your eyes again. So that was about half of you, maybe a bit more. So questions? Emma.
So for negative feedback, we said that T4 is converted to T3 through
Deiodinase?
Yes, and then it inhibits TRH?
Right.
I think that's number 8 in the top right corner.
Oh, down.
Down. Yeah, and then that's like TR3 increasing metabolic rate.
So we have two different locations. So the question is about negative feedback and the difference between converting T4 to T3 for negative feedback, which is decreasing metabolic rate, versus converting T4 to T3 in other cells, which is increasing metabolic rate. The reason comes back to a general point for hormones, which is it's a message, but the cells don't have to all respond in the same way. Okay? So you could have, in fact, this is true for hormones in general, you can have one set of cells do one thing. For example, for fat cells, should they release fats or use fats? Okay, so for adipocytes, which store fat, in response to T3, should they use the fat or should they release fat? Okay, you want them to release fat. You don't need to increase the metabolic rate of a fat cell. When a fat cell has a high metabolic rate, one of the things it's doing is making fat and storing it. So you want to change that to be breaking down the fats and releasing it. Whereas in other cells, you want them to be using the fats, breaking it down if they have some, but a lot of fats don't have any fat storage. So you're upregulating enzymes that will use the fat to produce ATP. Okay? So same message. It's often said, one message, many responses. Okay? And in this case, the T4, when TRH neurons receive T3, it inhibits them. So it downregulates the production and or release of TRH. Okay? And that's because the place where the T3 receptor binds is for alleles that would ordinarily be allowing the production, but in this case it suppresses them. Okay? Whereas in other cells, the genes that are available for binding that have the right combination of other transcription factors are actually increasing metabolic rate. Okay? Katie?
So if both T3 and T4 are produced in the original cell, step 4, if both of them are produced, then why is T4 the one that goes to like the negative feedback and conversion if T3 is the one that actually...
Yeah, so the question is, if they're both produced, why is it that T4 is the one that goes back for negative feedback instead of T3, which is there, right? So, very sensible question. Why is it T4? So a reason for that is something that I haven't told you guys yet, that T4 is much more abundant than T3. Thyroid gland actually reduces much more. This is not a crucial point. You can make sense of all the rest of it without it. So if nobody had asked me this question, I would not have said this. But T4 is much more abundant. Therefore, the T3 actually does inhibit TRH neurons, but there's not enough of it to make much of a difference. You need the extra from the T4. So T3 alone is not doing much unless the T3 levels are sky high. But all that T4 can inhibit it. Okay? And so, in effect, another way to think about it is it's actually in TRH neurons. You wouldn't be wrong to say it's actually the combination of T4 and T3 that are inhibiting it, because the T3 can also do it. Okay? Okay, other questions? Gil?
The T4, or the conversion of T4 to T3 in the TRH neuron, is that something with a receptor, or is it like just block some channels and stop the ion?
So the conversion of T4 to T3 in those TRH neurons, and you're asking...
Exactly why is less TRH secreted?
Oh, so that why there's less TRH secreted, that's the events... Where were we? That's the events down here in the thyroid. So this is a thyroid gland cell. And the enzymes produced in the thyroid gland simply are making many more of the enzymes to make T4 than T3. And that's just the way the regulation of the enzymes for both of those functions. Okay? So animals in general, their thyroid gland cells are producing more T4 than T3. Does that make sense? And we haven't gone into it, but you could hypothesize that activation of the enzymes to make T3, some key rate-limiting enzyme, is simply never that fast. And so you don't make as much T3 as T4. And that's the first part, right, of your question? Except your brow is furrowed.
Yes, but also like what happens in that one neuron, like the TRH neuron, that stops that?
So what's happening? So the second part of your question is what's actually happening in the TRH neuron? That would be down here. So what's going on in that neuron? I don't actually know the answer to that question. You could propose something, right? So you could, in order to make TRH, it has to involve protein synthesis. Either protein synthesis to make the small molecule. In this case, TRH is a peptide, so it's a chain of amino acids. So it's going to take transcription and translation of that TRH gene. And if you just inhibit it, you're going to make less TRH. Okay. There's another way you could do it. You could activate or upregulate the production of, say, a potassium channel gene. So if you produce more potassium channels, you're going to make the TRH neuron have a little bit more potassium leave. It's going to be more negative. It's going to be harder to get action potentials. Okay? So multiple ways to do it. Okay?
For the exam style question, if we were asked to do something with SCAPE, like T3 and how it acts on cells, would we need to go through the whole process?
So for an exam style question on one of these things, would you need to go through the whole process? No, that would take a long, long time on an exam. So an exam style question, I would ask you something. I already have some in mind, and you'll see some old ones. And you will definitely see a question, at least one, on something having to do with this. And so I would ask you, here's a change, like here's a drug we've added, or there's a disease, or there's a tumor. We have more of these cells, fewer of these cells. We have a drug, or we have some environmental stimulus. It's really, really cold. How would that change the amounts of TRH, TSH, and all the thyroid hormones? Another one could be we've downregulated a transporter, or we've upregulated it. So we have more of the transporter proteins or fewer of them. Or we have more of a receptor or fewer of the receptors. What would that change? And if you can tell me how that would happen, that's fine. If I want you to talk about a particular mechanism, I'll say that in the question. Briefly describe this part of the mechanism or something. In the back, I think Tanner?
Is the DIO enzyme within the TRH neuron?
I couldn't find it. So is the deiodinase enzyme inside the TRH neuron itself? I checked for this last year, or 2 years ago, and I couldn't find whether it was or not. It's got to be known. Well, it could not be known yet, but this is a pretty heavily studied pathway, so I think it's probably known, but I couldn't find it, so I just put it outside. And it seems to be T3 that actually does it. So it gets converted to T3 before it's being used, and it's using the T3 receptor. Okay. And there was another hand in the back, I think, yeah.
Could you go back to the drawing where T4 is being broken down?
Okay, so here we are in the liver. Okay, so T4 is being broken down. Uh-huh. You just wanted the drawing? Okay. Other questions? Yeah.
So when cells like need to convert T4 to T3 because they have a higher metabolic need, since the thyroid hormones are like being excreted constantly and there's like only small amounts in the first place, how would those cells get any T4 if the overall body like needs a little?
Okay, so the question is, if you're constantly converting T4 to reverse T3 and getting rid of the T3 and T4, and there's not that much of the hormone, how is it that there's enough T4 left for the cells that are increasing their metabolic rate by converting that T4 to T3? Because you're constantly getting rid of it. There's a constant tension, balance between production and removal. And so as long as the thyroid continues to produce T4 and T3, that's balancing the T4 and T3 that are being removed. When you suppress metabolism, you decrease it, and it actually takes a while. It takes, for these hormones are some of the slowest to be cleared. It takes half a day or a day for them to adjust the amount. And one of the main reasons is there are all those binding proteins that are holding T4 and T3, and it takes a while to get all of it off. One of the things that means is that if you happen to have a period with more or less iodine, your body is a little bit buffered from having a rapid change. And for the most part, we don't need a really rapid change of metabolism. If you're too cold, mostly what we do is a short-term response. We put on a coat, we become more physically active, we light a fire or whatever, hopefully not in your dorm room. Katie? Yep. So for the excretory process, would it be the same for all hormones, or is this specific to these? One reason I chose this system is that almost everything that's going on here is going on for other hormones in one way or another. And for clearance, the two main ways to clear them are enzymes in the liver. Sorry, my amplified coughs are going out through here. Enzymes in the liver that convert the active form to an inactive form. And so on every pass through the liver, you're decreasing the amount of hormone. And then in the kidneys, in the excretory tubule, this would be true for any animal. In a mammal, it's a nephron in the kidney. There are gaps small enough to allow these small molecules in, and once in, they're not recovered. What I don't know for these is whether the iodine is ever recovered. It's actually possible that the iodine is recovered. Simeon? So what makes this more efficient than neural pathways? So great question, and another one of the things I want you guys thinking about is when should selection favor using hormones and when should it favor neurons? This is an ultimate cause question. Why use a hormone? Why does selection favor this? When you want to increase your metabolic rate, you typically want to increase your metabolic rate in all of your cells. So every muscle cell becoming a bit more active, repairing damage a little faster, maybe making new protein. You want every cell in your intestine to be active, every cell in your brain to be more active. And if you controlled metabolic rate using neurons, you would have to have an axon branch going out to every single one of those cells. Enormously expensive. So whenever the targets are many, many cells, selection tends to strongly favor using hormones instead of neurons. When the targets are few cells, selection tends to favor using neurons, but that's actually only when those few cells need a fast response. So neurons for responses that need to be fast and where there aren't too many cells. Hormones when responses can be slower and there are a lot of cells. There are some things that it would be great if they were faster, but it would be too expensive. So as part of your fight or flight response, it's the sympathetic division of the autonomic nervous system, something we mentioned briefly. as part of your fight or flight response, you're actually changing in every single blood vessel. You're changing whether you're sending more blood to muscles, heart, and brain. That's fight or flight. Whereas when you're sitting there relatively calm and inactive, you're sending more blood to liver, intestines, kidney, okay, maintenance kinds of functions. And that means you've got to either expand or contract every blood vessel. And you can't do that. It's too expensive to do that with neurons. So your fight or flight response actually takes a bit of time. Not long, it's still on the order of only a minute or so. But to really ramp up all that change in blood flow still takes a while. Zach?
Two questions. One, are all hormone binding proteins produced in the liver?
Okay, so are all hormone binding proteins produced in the liver? I can't answer that. I can tell you that many of them are. I don't think it's been checked for all of them. And there's a reason for that. The liver has a structure that makes it easier for cells to produce bindings that will wind up in blood vessels. Capillaries are leaky, but in most places they're not leaky enough for proteins to get in and out. So if you want to get proteins into the blood, most of that is done in the liver. Okay?
T4 bind to DNA to produce DIO2, whereas T3 binds to DNA in order to produce proteins and stuff like that. In order to produce the enzyme with T4 bind a transcription factor.
So T4 is largely inactive. It apparently can weakly bind, but largely inactive. T3 is by far the most important one. So T3 binds to the receptor, the T3 receptor, and that's what then is, and the receptor is a transcription factor, and so that's what's regulating other genes. And I'm not sure I heard all of your question. Did I answer it?
DIO2, how does it know how to produce more of that in order to, is it always producing it, or is it
So, right, so DIO2, that enzyme fits on T4 and it cleaves off the specific iodine that makes it active T3. And so how do you regulate that? The cell needs to either increase the amount of DIO2 it expresses or decrease it. Or it could also be, a lot of enzymes are activated or inactivated by putting a phosphate on them. And so it could be that a kinase is putting a phosphate on it to activate it, and a phosphorylase is taking a phosphate off. But in any case, you need to change the amount of available DIO2, and that is regulated by something else. So if a cell's need to have a higher metabolic rate, then some signal is indicating, is activating a second messenger or activating a transcription factor, which would produce, for example, more DIO2.
But that would not be T4 activating that?
It wouldn't be T4 activating it. No, it would be some other signal. Like in the case of an infection, some immune system messenger molecule might bind to a receptor and activate the production of more DIO2. And then those cells would produce T3 and become more active. In the back?
So I have two questions. So the first one is that earlier in this process you had DIO, and now it's DIO. What's the difference between DIO and DIO2?
So DIO is just deiodinase, and you don't need to remember the difference. DIO just stands for deiodinase, and there's the different ones. So there's DIO2, which does this, and DIO3 converts T3 to T2. Okay, converts it to an inactive form. It takes off a different iodine. It also converts T4 to reverse T3 by taking off that same different iodine. You don't need that detail. Okay, I want you to know that happens, but
And secondly, I'm kind of confused on how the T4 and T3 enter the cell, because you keep drawing channels. But
transporters.
Transporters.
Yeah.
So if they're not water soluble, why can't they just pass through the lipid?
So that's the one way in which this is a weird hormone. I've actually tried in talking with some people who study this. So Professor Allison here studies this particular hormonal system. And what I've been able to read is that it looks as though at least some T3 and T4 may be able to just cross the membrane passively, but it looks like most of it needs this protein transporter that changes conformation and moves it in. Okay. I have not myself been able to get a clearer answer than that, and so I'm not sure to what extent it's simply the kinetics are not well worked out, or I just don't happen to know it. But it's water insoluble enough that it could conceivably, significant amounts could get across the membrane, but that's clearly not the only way it's doing it. Okay, and that's Now, every other hormone we talk about, it is either water soluble, in which case it can't cross the membrane and it's binding to a surface receptor, or it's lipid soluble, and it can cross the membrane easily and it binds to a transcription factor on the inside. Okay. I think Liam had a question.
After T2 and T3 get into the blood, are they just excreted or can they be converted back?
So right, so reverse T3 and T2 never get converted back. So once they're converted to that, they're now inactive and they're just excreted.
And then do they become, will they eventually be excreted?
Right, so they get into the developing urine, the excretory fluid, and then just wind up going out in the bladder and so on. Brendan? Yeah.
So does the cell take in just the hormone or the binding protein?
Does the cell take in just the hormone or the binding protein and the hormone? Just the hormone. The protein itself, pretty big. It's not that easy to move proteins across a membrane. So secreted proteins, they're either in a vesicle that pops open, or some proteins are moved out by actually a pretty big protein structure called the translocon. You don't need to remember this. But it's a fairly big complex of proteins that apparently can bind proteins and move them through what amounts to a sort of a huge channel that fits the protein and allows only the protein out. And those, so proteins don't easily cross membranes. Okay, one more question and then I think we'll move on.
Sort of along with that, how does the thyroid hormones like get detached from the binding?
So how do the thyroid hormones get detached from the binding protein? So that one, I'll remind you about the distinction between specificity and affinity. So specificity, it binds that and only that, high specificity. Low specificity binds multiple things with similar shapes and charges. Versus affinity is how tightly it binds. So for a binding protein like this, selection should favor reasonably high specificity if you don't want it to bind other things, but not really high affinity. Really high affinity and it'll never come off. You want some level of affinity where they'll pop on and off at a pace that matches the use for them. So selection should favor, for some things, low affinity. You want them to bind for a little while but pop off quickly. In this case, you need to hold them in the blood for a while, and so it's probably a moderate, not a high affinity, but a moderate level of affinity. Okay. So I posted a question for you guys that is the sort of thing I could ask on an exam. Propose a way for glucose or cold to change the amount of TRH hormone release. So for each of these, I'm not going to work through the whole problem, but for each of these, what I'd want you to be doing is going, okay, I know how thermoreceptors work. I know how G-protein coupled receptors work. I could make the connection to change what's going on inside the cell to either block action potentials, reduce action potentials by, say, opening a potassium channel, or by making action potentials more likely by either making more voltage-gated sodium channels or having fewer potassium channels or blocking potassium channels, so multiple ways. Okay. And for cold, doing something similar. I had another question about how could you have thyroid hormone enhance some genes, in other words, more of some proteins, but actually reduce the activity, reduce what's going on with others. And that gets back to deciding that for a particular gene, T3 binds to either an enhancer, in which case you'll get more gene expressed and more of that protein, or a repressor. And if a gene has that binding site for the transcription factor, if it's in a place that gets in the way of RNA polymerase, it's a repressor, and you'll block transcription. But if it's in a location that makes it more likely RNA polymerase will bind, then it will enhance. Okay, I'm going to talk occasionally or send you guys some emails about career-related things. Show of hands, how many of you have been to the Cohen Career Center? Great. For the rest of you, you should go. Show of hands, how many of you have developed a 3-semester plan to improve your skills, abilities, and so on? Not so great. All right, I'm going to send some stuff around. Use the Career Center more. It's incredibly useful. I have seniors, worked with one in my lab this year who got to senior year and wasn't sure what she wanted to do, went to the Career Center, talked with them some, and there's a career she didn't even know existed, and hopefully she has a job offer now. Don't know yet, haven't heard. And otherwise she would be going into next year thinking, I think I need to take a gap year. Okay, and she doesn't need a gap year. Okay, so I'm going to send some stuff around. I'm also going to have Professor Swaddle talk about one of the kinds of things you can do, and I'll send some other messages around about this too, in order to develop your experience.
Good morning, everyone. I think some of you know me. I'm John Swaddle in the biology department. I'm here to recruit for a research opportunity. Every year for the last 5 years, I take a couple of William & Mary undergrads to Australia for a field ecology, behavioral ecology research experience. It's 9 weeks in Australia, in the field largely, fun chasing birds, doing behavioral ecology type work. The commitment from the students is a whole year. What we do is, this is a program that's run by NSF, where we're in collaboration with a couple of other universities, Tulane and Cornell. And in the spring semester before the summer, we develop proposals based for the summer fieldwork. In the summer, we actually go and do it. And in the fall semester, you write up what you did over the summer. And from my perspective, the students who get involved in this at William & Mary, I actually expect students to stay in the lab longer than that afterwards and continue, whether it's that particular work. Unfortunately, there's not another opportunity to go back to Australia after that summer, but there's opportunities to follow up on that work, either in other kinds of projects in the lab or something that's very much related. So what you can do to find out more information is I can try and find a pen and write my email address down. Ooh, he's fancy. That works. I'm not going to break it. Okay. So this is my email address, John Swaddle. If you've not heard of me, I have a ridiculous last name, so you'll remember that. If you want more information about potentially going to Australia, I should say also that for the summer work, all the costs are covered by NSF. So this is, you even get a small stipend while you're there. So you're not out of pocket for going to Australia and doing this research experience. So if anybody's interested, email me and I can send you more information, including there's an application. But unfortunately, you have to be a returning student. So unless Professor Heidemann is going to enroll in some other discipline at William & Mary, then maybe he can go. But I don't know if he makes the grade. I think you guys are probably better at doing this. Then let me know about it.
Okay, so I'll be sending you some more information and things. You'll see a few emails and things, and I may bug you again about going to the Career Center.